Hey, everybody, what's doing? Mr. Yachts. Day two of notes covering cells. So we're going to go over some cell organelles today and some cell membranes, stuff you've probably been taught in junior high science, I'm sure, but it doesn't hurt to kind of go through it a little bit more again and review and maybe add a few things here or there. So let's just jump right in. So let's talk about cell membranes. Cell membranes, yeah, they obviously separate the cell from other cells. They're a border, okay? But the big thing is they're semi-permeable or selectively permeable, which simply means that they can allow some things in and some things out, but they're selective how they do that. And that protects the cell from injury and it just controls what's coming in and out. And they do that by structure. You'll hear me say this a lot in biology. Structure determines function, and you can see we're going to use some terms from some previous chapters. Lipids, which you guys remember from the macromolecule notes, those are fats. And then we have these big boulder proteins stuck in that fatty layer, bilayer. And basically, if you look at this picture, it only allows certain sized molecules to pass through. That's how they're selectively permeable, both directions. This is the inside of the cell. This is outside of the cell. And we can be very, very selective what comes and goes. Kind of interesting how this bilayer is composed. These red head-like structures on both sides, those are called hydrophobic. If you know of someone who's a hydrophobe, they fear water, so they repel water. But then these tails are hydrophilic, which means they attract water. They love water. So you can see the spaces between them, and that's where things flow. And that's what makes a cell membrane or plasma membrane. It goes by both names, kind of interchangeable. Good A/B question on the test. That's what allows us to be selectively permeable and allow things to move through a cell. And that's what they show here too, the green and the blue inside outside cell. That's what protects us. Now I know there's bacteria, there's viruses, there's things that have figured out if I'm small enough, I can still slip into that cell and cause some havoc, and they do. But for the most part, our cells do a pretty good job of protecting us from harm. Okay. Now, a plant cell has an additional layer on the outside. Now, be very careful on the test because they also have plasma membranes, just like we do, cell membranes, but they have an extra layer that's not living on the outside, and that's called a cell wall. And that's what allows plants to get 30, 50 feet tall and withstand wind and environmental conditions. They have this extra layer. So if you look at a plant cell, let me show you that. They have a plasma membrane. There's the outside border. That's where our cells in. Then they have all these extra layers on the outside, sometimes wood, sometimes bark, sometimes just stemmy material. But that's what gives a plant its stability and structure, which is a little different than us. We have a body and a skeletal system that can protect us. So that's what you see when you look at plant cells. And plant cells have cell walls, animal cells do not. However, both cells have plasma membranes. Okay? Now, let's kind of change gears a little bit. Let's talk about some organelles. The question for Thursday was, that was the answer. What's a tiny organ? They're called organelles. So let's start with the nucleus. These are eukaryotic cells. Only eukaryotic cells like us, plants, animals, fungus, they have a nucleus. A nucleus is where we have our DNA, okay, and a second membrane called the nuclear membrane. It's a large oval spherical body near the center. The key word there is near the center. It's not exactly in the center, even though your diagrams a lot of times put it there. It's just towards the center. If we look at some cells that we looked at in lab, you can see these circles in these rectangles in a plant cell. Those are the nucleus. Okay. Now, a nucleus does several things for us. It's kind of the brains of our cell. But what I really want you, it controls all cell activity. What I really want you to understand is what is inside of the nucleus, what organelles are inside of that nucleus. So this gets a little confusing. So we have a circle in a circle. I know that sounds weird. Wheel inside a wheel. Think of a hub in the center of a big wheel. Okay, that's called the nucleolus. have a lot of students that get nucleolus and nucleus backwards because it's also an oval object, but it's inside the nucleus, and it controls protein synthesis. Okay, and it does some other things for us as well, but the main function is control protein synthesis. If you look at your coloring sheets, which I hope you guys have done, I'll show you a picture here. That's the nucleus. The outside one, the blue circle inside there is the nucleolus. And then we have these little gummy worm things, okay? And I don't know why the people that study cells back in the day got stuck on the letter C, but they did. So there's a lot of C words. I apologize for that up front. There's just a ton of C words in this chapter. And we have two of them here. We have something called chromatin and chromosomes. You're either going to have one or the other. Chromatin is what you see most of the time. That little gummy worm stuff I just showed you is unorganized active forms of chromosomes. Chromosomes only appear, they're like little X's. They only show up when a cell's dividing. So you don't see them very often. You only see them when a cell's going through cell division. And that's about the only time you see that. Let me see if I can find a picture of that for you. Think of, while I'm trying to find that, think of a bedroom. I'm sure all of you guys are nice and neat and everything's put away, but if you're anything like my daughter, looks like a bomb went off in her bedroom sometimes. That's what you see with chromatin. It's unorganized. It's just clothes and stuff thrown everywhere. Chromosomes are organized forms of DNA. They're like little X's. I don't know what happened to my picture. I'll see if I can still find it here. I don't know. Somewhere on there, I probably passed it. But, that's what you see, the difference between the two of them. Usually only seen when you're only going to see that when cells are dividing. I don't know what happened to my picture. Sorry about that. My links I usually use, but when I'm doing screencastify, I can't use the links. So here we go. Finally found it. So this is chromatin, all this little unorganized black stuff, and then eventually gets organized and becomes these little X's called chromosomes. So that's what you find in a nucleus, chromatin, if it's 90% of the time. And about 10% of the time, you will find chromosomes when cells are going through mitosis and meiosis. All right. Couple other things. I want to go over two more slides, and then we'll be caught up with notes. Inside of a cell's cytoplasm is a gel-like structure. You don't really see it under the microscope because it's clear when the light shines through it, but it's actually, if you would break it down, it's colloidal, which means it has a gel-like substance to it. And that's why it kind of holds things in place. So inside the cytoplasm is the endoplasmic reticulum. There's a rough and there's a smooth, okay? They do the same thing. They're a canal system. I used this analogy in my class yesterday. If you fill a room full of Jell-O and you had to walk across the room, it would take you a while. Sometimes we got to get things into the cell super fast, and it doesn't have time to wade through the Jell-O. So what we do is we put the endoplasmic reticulum in there, which is a system of canals, rough and smooth. Let me show you a picture, okay? And you can see the little canals that run through there, and that way things can pass through the cytoplasm by bypassing. I put a pipe in that Jell-O, room full of Jell-O, I can walk right through, and that's what we do. The only difference between rough and smooth is rough looks kind of grainy because it has ribosomes attached to it. Smooth does not. So that's the big difference between the two of them, even though they do the same thing. So don't be confused about that. They're involved in the same business. They just have a different looking appearance to them. Okay. And those ribosomes that are attached to the endoplasmic reticulum, they're grainy, they're small, but don't be deceived by them either. They have a very important function, and that is protein synthesis. Okay, and think of the cell as like a factory. We make cars and trucks and campers and things like that. Maybe some of your parents work in a factory like that, where each person does their job, and then by the time we get to the end of the line, we have a finished truck or car or camper or whatever. That's what the cell's like. Each part plays their role, but one part's not more important than another. They all just have to do their thing. And ribosomes fit that bill. They synthesize proteins. We'll talk more about them in some chapters coming up. The mitochondria, you can see what the main function of mitochondria are. They're a center of respiration. 95% of the energy needs of a cell is met with the mitochondria. They also control calcium and water and some other things coming in. But the main function, I'll show you a picture of those, is to control or is to provide energy for us. And this is what they look like. They have a double membrane. They actually have their own DNA, which is kind of cool. These are your brain cells. And I asked my class a question. I had some different answers to this question, but eventually they came up with it. It was what organ in the body would have the most mitochondria. So think about something in your body that would need a lot of energy 24/7. That would be your heart. So cardiac muscle has a ton of mitochondria because, and the brain does too, second one, because we need so much energy in those two organs. Okay. The last one I'll talk more about in the next set of notes. That's called the lysosomes. They're called suicide bags. That sounds like a weird term, but they're bags of enzymes that when a foreign thing comes in the cell, they explode, they sacrifice themselves, and they dissolve and get rid of whatever foreign body that is. So they're the protector of the cell. Not real big, but they're scattered all over the cell, and they do a great job of defending and getting rid of injurious things. I'll go over those a little bit more in the next set of notes. I just want to kind of introduce them here. They're not real impressive looking under the microscope. They're just these little round structures. You can see them kind of scattered throughout, but they're very, very important in the protecting of our cells. All right. So not a ton of notes today. Just kind of wanted to start our organelles. We'll finish those up on Monday. I'm not going to probably have a test until after fall break. So we'll just keep going through notes, and hopefully we can get them finished by Wednesday of next week, the short week. And then when you come back, probably on a Tuesday, we'll take our test and get it in for the end of the trimester. All right. Hey, have a good day. Hope see you at Google Meets later today, 12:45 again. Don't forget, and we'll pick up there another day. See you. Have a good day.